Good morning, everyone. A very warm welcome to the Institute of Food Science and Technology's Food Safety Group's webinar, Foodborne Viruses and Emerging Issues. We've got a great speaker with us today. I'm Sterling Crew and I chair the Food Safety Group. Just if you have some questions, can you hold them to the end? You'll see down on your screen a Q&A button, put them on there, and we'll try to get through as many of your questions as we can at the end of the presentation. It makes a great pleasure to introduce our speaker, Martin D'Agostino, who is the virology manager at Camden BI, a very well-known figure in our industry and is an eminent person in his field. So Martin's specialism and expertise includes the investigation of transmission of enteric viruses through various supply chains, including norovirus and hepatitis A through the fresh and frozen produce supply chains, and hepatitis E virus through the pork supply chain. Martin's been involved in several of the really large EU framework projects in the past, involving the development of rapid molecular methods for the detection of foodborne pathogens, including projects related to methods for the detection of viruses, both in environmental and food scenarios. So without further ado, over to Martin. An interesting time to be a virologist. There seem to be so many of them now coming out of the woodwork with COVID-19. But for today, it's about 2 out of 3 foodborne poisoning cases are caused by a virus. I never knew it was so high. So viruses are a significant challenge to food safety. Martin, over to you.
So hi everyone, thank you very much to Sterling and to IFST for the opportunity to present this information to you today, and hopefully it will be an interesting discussion, question and answer discussions as well that we can have after the presentation. So Sterling mentioned we are going to be concentrating today on talking about foodborne viruses and emerging issues from these types of viruses. So covered today, we'll be having a look at a brief introduction and some thinking points to ponder as we go through the presentation. What are foodborne viruses? So just a reminder for those of you who are not that familiar with these types of viruses, what are they, and what size are they, and what do they do? The transmission routes of foodborne viruses. What happens when viruses are found in foods? So a couple of look at a couple of outbreaks and RASIP reports related to foodborne viruses. Discussing some issues surrounding foodborne viruses and what lies ahead in terms of the future with foodborne viruses. So just as a brief introduction, I just wanted to introduce some just general information regarding how foodborne viruses are viewed by the food safety authorities. So back in 2015, the Advisory Committee on the Microbiological Safety of Food, they produced a report back then and specifically mentioned viruses in this report. And the conclusion was that especially berry fruits and leafy green vegetables, which have been implicated in several outbreaks of norovirus gastroenteritis globally. These are issues which are of a high rank in terms of risk ranking, in terms of the types of produce that are associated with contamination with viruses. Also, the European Food Safety Authority, their Biohaz panel, back in 2013, also risk ranked the combinations of leafy green vegetables with norovirus, raspberries with norovirus, and strawberries with norovirus as having a high importance for human cases of infection originating from food of non-animal origin in the EU. But also the UK Food Standards Agency recently published new estimates concluding that about 380,000 cases of noroviruses linked to food, and that occurs each year in the UK. So there's lots of evidence to show that these issues have been around for a long time. Regulatory authorities have known of the issues for some time. There's been lots of research work performed on these types of viruses and their association with foods and how they can be detected. And as Sterling mentioned, I've been involved in quite a few of those larger projects to date. So it's not a new area. Some of it's emerging, which I'll come on to, but it's certainly been known about for quite some time now. So some things to ponder. Why should the food industry be concerned about viruses? Things like the presence of norovirus and hepatitis A and E in our foodstuff. There isn't any microbiological criteria currently in place. They don't fall under the same regulatory laws as, for example, Salmonella or Listeria do, unless there is a temporary mandate in place, which there has been in some cases. For example, frozen strawberries from Serbia, for example, have been mandated to be tested when they come into the country. The most common cause of foodborne gastroenteritis globally is caused by norovirus. And there have been several published reports from different countries showing that norovirus is at the top of their lists of foodborne illness cases. And could that be linked to the above? Perhaps. It could be a case of the fact that no one's really got anything in place to monitor for these viruses in foods. But equally, there's maybe some assumptions in place, which we'll also be discussing a bit later on as well. And then also there's the human interface. Your workforce, visitors, perhaps customers who come into your premises, they all have the potential to spread contamination to foods and surfaces. So these are transmission routes, which we'll also be discussing a little bit later. There's no compulsion to look for them or monitor for them. So contamination events could be easily missed. What's your view on these kind of things? Maybe you have a couple of questions regarding that, or even comments, viewpoints about that. But these are certainly areas why I believe the food industry should be concerned about viruses. Also, many outbreaks have occurred, both internationally and nationally. Some of them have been very high profile, not all of them in the UK, obviously. There's been a lot elsewhere, but there have been a few in the UK. Since there are no current regulations in place requiring the monitoring of these viruses, does this mean that food business operators shouldn't consider viruses as risks to their businesses? But if they did, what actions are taken by those businesses? And if there is an issue, what are the issues preventing food business operators from undertaking regular monitoring for viruses? Could it be a lack of data? Could it be the fact that they don't know if they're infectious or non-infectious? Could it be the cost of looking for these? Could it be the not knowing where to look or not knowing what actions to take if positives are found? Or is there just a lack of interest? So just moving on slightly from those thoughts, should viruses be considered as hazards in food safety management plans? Well, back in 2011, a whole 10 years ago now, EFSA's scientific opinion looked at norovirus and hepatitis A viruses in fresh produce, ready-to-eat foods and bivalve mollusks. And these include things such as oysters, mussels, and scallops, as those were ranked as priority hazards by the World Health Organization. Hepatitis E virus was also assessed in the opinion as it is highly prevalent in pigs across Europe. And that situation certainly hasn't changed at all. The ACMSF report of 2015 mentioned earlier, that was titled An Update on Viruses in the Food Chain, concluded this: the contribution of contaminated fruit and vegetables to foodborne norovirus and hepatitis A virus is uncertain, but the impact at population level could be significant given the consumption levels. Protection of the consumer relies on adoption of and compliance with non-statutory hygiene schemes. So bearing all that in mind, we know that there's an importance and a recognition of foodborne viruses in the food industry. We know that there's issues associated with them. We know that these have been given a lot of consideration. So just before we discuss any of those points further, let's have a look at the types of viruses that are of most importance, first of all, to the fresh produce sector. So for this, we're discussing norovirus genogroups 1 and 2. Other genogroups are, of course, in existence, which are associated with different animals, but these particular ones are associated with spread through humans. Hepatitis A virus and hepatitis E virus. There's a question mark over that because there isn't a whole lot of work being done on hepatitis E virus detection in fresh produce, or even if there is an actual link between the possibility of getting hepatitis E in fresh produce, but there is certainly a possibility for that transmission route to exist, which I'll come on to in a moment. So what features differentiate viruses from bacteria? Well, firstly, they cannot reproduce like bacteria can outside of a host cell. They are able to remain infectious through the freezing process. So that means that they can potentially remain infectious for long periods of time. And in fact, this is how we store viruses in the laboratory. They are able to be transmitted by foods and may remain infectious for a considerable amount of time. Of course, particularly fresh produce, of course, has a relatively short shelf life. So it's possible for these to be transmitted through these foods right up until point of consumption. They are very small. They're about 25 to 40 nanometers typically, about the 100th the size of bacteria. Approximately 18 virus particles might cause norovirus infection in a healthy individual. I'll go into a bit more detail about that in a moment. And there's a lack of robust cell culture systems in place. It's very difficult to directly investigate the effects of potential process control measures such as heat and pH, etc. But a lot of the produce which we're talking about here do not get heat treated anyway. We're talking about things like fresh leafy green vegetables and soft berry fruits. So looking a bit more closely at foodborne virus transmission routes, how do they actually get from the person into the foods? So the food chains implicated in outbreaks have mainly been associated with fresh produce. So again, we're talking about leafy green vegetables, soft berry fruits. This can be from a number of sources, potentially from contaminated handlers' hands. It may be from irrigation water, which has become contaminated with potentially raw sewage, or water used for pesticide preparation, which may not be potable water. So when we think about irrigation water, we know that a lot of crops, of course, are not necessarily irrigated with river water, but a high percentage are. And there's still a lot of crops in the UK which are irrigated using water directly from rivers. So if there is potentially, for example, a flood scenario, there's been lots of rainfall and sewage outflows become so great as they discharge raw sewage into waterways, that can spread down to the irrigation systems for these products downstream fairly easily. Again, also water used for pesticide preparation. If you're not using potable water and it happens to be contaminated with viruses, you could have a way of spraying these viruses directly onto the surface of the crops in an even sort of fashion. So that's another one that's been identified as a potential for contamination. Frozen berry fruits via handlers' hands, again, irrigation water processing in the patching areas could be an issue. Shellfish, of course, is a big one via contaminated water. If they are harvested in an area where sewage has been discharged, obviously these shellfish are filter feeders. They can filter out larger debris, but viruses can accumulate within them. And then we have hepatitis E in pork products. Pigs are infected with hepatitis E. 93% of the pig population in the UK is endemic with hepatitis E. So these have the potential to spread from pig farms to watercourses, which can then potentially go into irrigation of other products. So just as a brief sort of diagram, just to illustrate that, in the middle, of course, you have the human, the source of these particular norovirus and hepatitis A virus. How do we excrete them? In our stools and potentially in our vomit. If we have an infection, we can be contaminating toilet surfaces, toilet door handles, surfaces, etc. This can then, of course, go into our sewage treatment, into the sea, into river courses, groundwater, recreational waters, also all sorts of areas. Then, of course, they can go into irrigation water, pesticide preparation water, that can be applied to vegetables, fruits, and go into shellfish. Food handlers' hands, of course, are another potential contamination route. Food surfaces, food prep surfaces, and cooked foods. So anyone, an infected individual who doesn't have good hand hygiene may be the source of that contamination. On the other side, we've got, of course, aerosols produced by perhaps someone who's vomited. That can go into hands and clothes and be an issue, and that can go spread back to and other people in the area. And then the biggest cause, of course, of norovirus infection is down to person. We all heard of it. It's called the winter vomiting disease. It's known as the to be biggest concern between family members. In nurseries, it happens a lot. Care homes, hospitals, food handlers, restaurant areas. All these areas where people are in close contact with one another, and who maybe inside a lot more, especially during winter time. So we mentioned aerosolized particles can contaminate large areas within restaurants and food outlets and supermarkets. This picture on the left is an experiment which was done to find out how far particles of vomitus could be ejected during this type of an incident. Not the best of subjects, but you can actually look this up on YouTube. It was done by Public Health England laboratories, and this one's called Vomiting Larry. So very interesting there. They used fluorescent material to be able to show how far these particles could travel. But just imagine this happened within perhaps a supermarket or a restaurant. These particles, and depending on the airflow, etc., they could travel for fairly large distances. And when they do land on surfaces, the particles may remain infectious for many days, possibly weeks, depending on where they land. So just going back to how contagious norovirus actually is, we know, as we've already said, that low numbers can cause illness. If we're thinking of just about 18 virus particles potentially making you ill, this diagram on the left just shows that if you could fit viruses onto the head of a pin, you'd have enough to be able to infect more than 1,000 people just with one pin. So it just gives you the idea that it's very small amounts we're talking about here. You wouldn't even notice perhaps any soiling in your hand, which could potentially have norovirus on them. And it's estimated that ingestion of about 10 virus particles could result in a 30% chance of infection, and 100 virus particles could result in a 50% chance of infection. And that's one of the reasons why norovirus is one of the most contagious infections known to man. It's important to note as well, though, that many individuals may be asymptomatic. And you may have heard that term being used quite a lot more recently because of the SARS-CoV-2 pandemic. Asymptomatic, of course, means that you could be harboring and transferring the virus in your environment without actually having any symptoms at all. If you do have this kind of incident in a factory area, a processing area, ready to eat area, a restaurant, produce aisles, etc., clear guidance is required to be able to deal with this type of an incident, not only to protect the patient, but also individuals nearby. Cleaning instructions, equipment, personal protective equipment should be considered. You know, if there is a vomiting incident, you don't know what's caused it, but it would be wise to consider norovirus as the potential cause, just to be sure. So it's recommended that a 50-foot diameter around an incident should be cleared of products on sale in processing and production settings. And we do actually have guidance on how to deal with this type of incident on our website. What about hepatitis A virus? Well, prevalence and transmission. In the greyed out areas here, there's a very low amounts of prevalence of hepatitis A. We're talking more about Middle Eastern areas and Asian areas, South American areas, where the prevalence is high in the community. So these are places where hepatitis A is endemic. The problem happens when you have products which may be produced in these different countries, and they are being exported to the UK. So we are, you know, for example, if you go down to your local shop, you may well find blueberries from Scotland being in the produce aisle. You may also find blueberries from Chile or Mexico being there as well. So depending on where they've come from, you just don't know if they're necessarily from a high prevalence area or not. But this is the reality. We have a global supply chain where these products could be coming from anywhere, with standards not necessarily as high as our own in terms of hygiene, and possibly with people who are contaminating these products, and they're coming into the UK, but it's an unknown. Potential transmission routes for hepatitis E virus. Well, we've mentioned pigs. Of course, hepatitis E can be found in a number of various animals, but the most prevalent one is in pigs in the UK. Their waste may go into watercourses, can either be also direct contact with farmers who are pig farmers. Of course, that doesn't necessarily mean a huge amount of transmission takes place for them. Biggest potential is for the waste going into river water and potentially being put onto things like leafy green vegetables and us eating them straight afterwards, or the consumption of undercooked pork products. So hepatitis E is quite different from norovirus and hepatitis A in that norovirus and hepatitis A is normally going to be found in the surface of products, whereas hepatitis E is going to be within the actual meat itself. So foodborne viruses in fresh produce, just to remind yourselves, are unlikely to have had any heat process. They're obviously mainly eaten raw. The detection of low numbers of viruses directly from foods is required. They're not infecting the foods. There's no obvious deterioration of the food, so no replication on the surface. It doesn't impair the look or smell of the food, and it's not likely to be homogeneous contamination. Why is it prone to contamination? Well, we have to think about the microscopic structure of these foods. There's very high electrostatic bonds which attach the surfaces of the foods to the viruses, and they can stick quite strongly. So they do play a significant role in the attachment and subsequent difficulty in removal and detachment of the viruses. So just washing with tap water can get rid of a large lot of debris, but may not be sufficient to remove viruses. And we have talked obviously a lot in the last 18 months about SARS-CoV-2, but I just wanted to make one point clear that obviously good personal hygienic practices, good agricultural practices are key in the food industry for control of viruses. Good hand washing techniques are obviously required. And with the SARS-CoV-2 pandemic, of course, we've heard a lot about hand washing. but we've also heard a lot about alcohol-based hand sanitizers. Now, these are effective against SARS-CoV-2, but they're not effective against norovirus. So that's just one thing to bear in mind. Your key control measure for any of these things should be washing your hands first and using hand sanitizer as a secondary barrier. So some notable reports of viruses linked to foods. Back in Germany 2012, there was a largest recorded outbreak caused by norovirus. There were 390 institutions across 5 federal states which reported about 11,000 cases of gastroenteritis. The cause was found to be frozen strawberries imported from China. We don't know the cause of the contamination, but there was an EU mandate to test 5% of imported Chinese strawberries. Now, if you're familiar with Chinese farms, there are thousands of them which produce fruit, and they all get put into large distribution centers, and then they get sent out. So trace back of these things is very, very difficult. That's one of the main issues with regards to produce from these types of areas, is actual trace back to be able to get to the root of a problem when it occurs. We had one in the UK in November 2016, a well-known one, the Oaxaca restaurant chain, where 160 customers and 200 staff became ill. 18 of 25 restaurants were hit, with 9 forced to close temporarily. And the lawyer secured payments in excess of £15,000 for those left seriously ill, because it wasn't just a case of, a couple of people getting ill for a couple of days. Whether if you have underlying illnesses, in some cases it can cause bigger complications. And this was all linked to new menu tasting, which was launched in all restaurants. And it was possibly from an ingredient which was sourced outside the EU, which didn't have controls in place on it. So yeah, they had a big problem with that, and they lost a lot of money from that. But that just shows some of the impacts that it can have if we're not careful. A very recent outbreak has just been published in Food Safety News, norovirus outbreak in UK and Hong Kong linked to oysters. So the report suggested at least 100 people became ill in the UK, with 12 cases in Hong Kong from raw oysters produced by Whitstable Oyster Company in the UK. So yeah, these things survived. They survived that whole travel to Hong Kong, and they went on to infect people there. So it just shows you the resilience of these types of viruses, and they don't have any problem traveling around the world. Another recent report, this time from RASIF, was notified on the 16th of April. This was noroviruses in frozen raspberries, and it was notified by France, where they do a lot of testing of products for viruses in their foods over there. So that was another one. It's showing, again, as I say, freezing it doesn't cause any issues. It will preserve the virus on the surface of the foods. There was a big hepatitis A outbreak in 2013. Back in May 2013, there was a European alert regarding hepatitis A outbreak in Italy. So there was a big case control study done, questionnaires, generated data, and the hypothesis was the illness was associated with the consumption of frozen berries and possibly fresh blueberries. They selected food samples, but they didn't detect any. However, traceback investigations focused on imported berries to a single Irish distributor of imported frozen berries used in 9 of 14 cases. 12 cases were hospitalized. Thankfully, there were no deaths. But there was a boil order issued 1 minute prior to eating, was advised. And I believe that is still in force. That was issued by Food Safety Authority of Ireland. And I think that boil order is still in force to this day. In terms of other outbreaks, largest outbreak in the US was sourced to spring onions in 2003, where there were 3 deaths associated with that one. Largest outbreak in Europe was associated, as I say, with frozen berries in 2013, so 1,500 cases across 12 countries. And a large outbreak associated with consumption of raw scallops happened in Hawaii, 206 cases with 51 hospitalizations. But it just gives you an idea of the scale of these things. This year also, there was an issue with hepatitis A in dried tomatoes from Turkey that was found. Again, notified by Greece, and that went into the RASIF alert system. But over on the right here, we see that there was also a hepatitis outbreak associated with the consumption of dates in England and Wales just very recently. And that led to Sainsbury's actually recalling Taste the Difference medjool dates because of possible contamination with hepatitis A virus. 29 cases were identified by Public Health England, and within that Eurosurveillance report, it just mentions 2 cases there where confirmed presence of hepatitis A was found, and we actually did that work for Public Health England there. So what are some of these surrounding issues of foodborne viruses? We've seen that they can contaminate many types of foods. It can affect many different people. It can travel the world. So what are some issues surrounding these viruses? Well, as we've already mentioned, there aren't any real regulations in place. There's no microbiological criteria set for viruses in foods due to a previous lack of prevalence data. We do, however, have a lot more prevalence data now. There's been studies reported and published not that long ago. So that means that more meaningful control strategies could be developed. But there's also some assumptions and risks. Why is there a need to monitor for the presence of pathogenic bacteria, but not for viruses? That's one question to ask. The assumption may be that noroviruses aren't really that dangerous. The infection clears up fairly rapidly, and they're relatively easy to deal with. But noroviruses do not cause as many deaths as Salmonella, but they do have a major impact on social costs. A typical norovirus outbreak may involve up to 50 people on average, whereas a Salmonella outbreak may be only around 7 people. So there's all these different factors and risks to consider. What's actually being done? Well, the Food Standards Agency's 2012 Forward Evidence Plan made as one of their policy objectives was to develop an evidence-based approach to managing risks from and controlling the burden of foodborne norovirus disease. And that study was the NOVAS study. That was there was a lot of work done with regards to the norovirus attribution and study, which was coordinated by University of Liverpool under Professor Sarah O'Brien. And as a result of that, the FSA recently reported more estimates of the societal burden caused by foodborne illness in the UK. The main points were that the financial burden due to 13 main foodborne pathogens is about £3 billion, and norovirus imposed the greatest burden with an annual estimated cost of 1.68 billion, followed by Campylobacter species of 0.71 billion and Salmonella species, non-typhoidal, of 0.21 billion. So great economic cost. Unidentified pathogens causing foodborne illness was around 6 billion. That's the headline figure, but a headline figure of approximately 9 billion. So 60% of foodborne illness cases were attributed to no specific pathogen. Could that be because they just didn't identify them? Could it have been norovirus? Could it have been hepatitis A? Could have been, but it's more likely to be caused by ones which are not able to be cultured, those which are not readily looked for. So we just don't know where these other cases are coming from. But they could also be from viruses that we don't yet know about, as far as we know. So what lies ahead? Potentially emerging issues related to viruses. We know, of course, about the known foodborne viruses. These are the ones that we just talked about. So what's going to change? When will change happen? What changes need to be put in place to reduce contamination? At the moment, no one's really under the compulsion to look for these things. What about unknown viruses? Well, look at the impact that SARS-CoV-2 has had on multiple food chains across the food industry. Even although they're not actually considered to be foodborne, there was a massive concern that they were actually foodborne. There was cases of it being found in chopping boards, on frozen seafood, that could have been spread to people potentially. Other viruses might appear that have similar impact, with a possibility of actually being foodborne. But we need to have the expertise and resource in place to overcome these challenges. There's not many people actually do this type of work, especially in the UK. SARS-CoV-2 pandemic highlighted the impact that viruses can have in our lives. We have heard the message that we will have to learn to live with it. But should we learn to live with foodborne viruses, which could be on our foods with the potential to make us ill? The UK, as we know, is no longer necessarily governed by EFSA as directly as it once was. However, we do still import many foods from the EU, and of course we export them as well. And each of these countries have their own rules and regulations, which may be different to ours. The FSA report indicated that on average, if you calculate back the figures that they provided, about 315 people may be infected with norovirus by eating lettuce every day. Is that acceptable? How many other food types may be contributing to the spread of norovirus? The study that was done was on those very specific products. But of course, if you want to know all the potential routes of contamination, you have to do a lot more monitoring to find out, to further identify the risks involved. So quite a few issues related to viruses. There's a lot of things we still don't know. What's out there that could produce issues in the future, and that's maybe something that you have some ideas of your own. It is an unchecked risk. Norovirus, as we say, isn't under regulation, yet it causes a lot of the foodborne illnesses. We are, at the moment, the only laboratory in the UK available to analyse fresh and frozen produce for norovirus and hepatitis A virus, and we are UCAS accredited. We haven't talked that much about detection of hepatitis E virus in pork products, but we do have methods to be able to detect these. There is no standard method available though, but so we are involved in a Food Standards Agency project, which is looking at the standardization for the detection of hepatitis E virus, and we're hoping to be able to put that through to international standardization at the end of it. So there's a lot of things to consider there. I haven't covered everything by any means, but hopefully that just gave you a flavour of some of the issues related to foodborne viruses, some of the challenges involved. And if you have any questions, I'd be more than happy to answer them. Thank you.
Thank you, Martin. That was a really super presentation. Certainly refreshed my knowledge of viruses and demonstrated how important they are from a food poisoning type of perspective. We've got a great opportunity to ask Martin some questions. There's lots coming in already. Please submit your questions using the Q&A button that you see on the bottom of your screen. You might have to move your cursor around to activate it. You can also vote, so there are a lot of questions coming in, and if there's one that particularly you want answered, please do vote on it and it will go up the list. So we're answering the questions you want. So perhaps asking our first question, Martin, some states in the USA, such as in Missouri, are introducing compulsory hepatitis A vaccine for food handlers. Do you see a time when compulsory vaccines will be required in the UK? And if so, for what?
Well, that's a really good question. I mean, it's possible, but I think a lot of the issues caused by the consumption of either zero processed or minimally processed foods, often from countries where hepatitis A virus is endemic, is the main problem. So unless the whole population is vaccinated, those who eat all those foods, just vaccinating UK food handlers probably wouldn't reduce that risk to the general public, as it's not a common disease here in the UK, and the risk of infection is really quite low for most people. But I guess if hepatitis A infections increased significantly, for example, and they were associated with the consumption of maybe locally produced and locally consumed foods, that may trigger an action to vaccinate food handlers here. But yeah, as I say, the situation is different in different countries. It may be more worthwhile to vaccinate the food handlers in the countries where we are importing a lot of foods from.
Thank you, Martin. Here's a really interesting question as well, where the questioner says, look, I know we're starting to use whole genome sequencing as a method of identifying variants of bacteria. We've seen it in some of the recent
Yeah.
listeria, and that this can be used for traceability. It's a great tool. And what the questioner is saying is, could we do this same thing for viruses?
Yes, quite possibly. I think the ready availability of lots of data for bacteria is obviously a plus point for all the traceability that they have for bacteria. Because viruses aren't monitored so much in the UK, we don't have really a big database of the viruses that are spread through the food as such. The PHE will have lots of clinical data and information on the sequences of the public health related noroviruses. But in terms of those that are traveling through food, unless you pick it up and monitor that and detect it in foods, that's a little bit more difficult, especially with regards to the low numbers of viruses that you have on actual foods, because as I said, that they don't grow in or on foods, and you can't enrich them in that same way as you can bacteria. You have to do direct detection. Then and the genomic sequence that you get from when you do the molecular detection of the viruses, you have to have a lot of product there to be able to then go on to sequence that, and it's a very low base pair number that you're actually looking for. It's very specific, so sometimes that can be an issue in actually having enough material to be able to do sequencing of these types of viruses. But yes, I agree. If they were more available, and we had lots of strains to be able to look at, and more data and more positive material to look at, then we might have a better idea of traceability within the country.
It'd be a fantastic tool, isn't it? Linking up
Mhm.
environmental swabs and product swabs to see actually where the infection might have come from.
Yeah, yeah.
Another good question here. Is the viral contamination of pork due to poor evisceration methods and then cross-contaminating to the meat? Or is it present within the cells, the flesh of the live animal? If it is due to the evisceration methods, why is this not seen with beef, which is more commonly cooked? And you could say is more undercooked perhaps than pork.
Yeah, that's another good question. Yeah, I think the standard of evisceration methods is very closely looked at in abattoirs. I don't think there's any issues with any of that, because it's all very closely monitored by food safety inspectors. But the hepatitis E virus can be found in the feces, in the urine of pigs. It can be found in the flesh of the pigs, and it's carried through the blood of the pigs. So, liver especially is more likely to be contaminated with it. But yes, those products which are minimally processed may have a higher likelihood of containing hepatitis E virus. But again, there's not a huge amount of data out there or monitoring going on to know exactly how much of a problem that is. But certainly in terms of beef, that's not so much of a problem because it's not known to infect cattle or cows. It's mainly an issue in the pig supply chain. And because that's so large, that's where the main issues come from. Although it can be found in other species like wild boar and other small game mammals as well, animals as well. So, but pigs by far, because they're the biggest, one of the largest consumed products in the UK. That's why there's an issue with that.
And here's a question related to perhaps your role at Camden. are viruses more difficult, expensive to test for than bacterial pathogens? So, if you're going to do some testing, is it more expensive, less expensive?
No, that's another good question. Yes, it is more expensive because a couple of reasons. You can't grow it up on an agar plate like you can Listeria, E. coli, or Salmonella. They're very easy to, those pathogens are very easy to grow up in a normal laboratory under fairly easy conditions. You know, stick them in an incubator and you'll start seeing colonies. You can't do that with these viruses. As I say, there are cell culture systems available for hepatitis A virus and hepatitis E virus, but they're not commonly used for the food side of things. You know, so they would have to be validated to be sure that none of the food components affect the growth of the virus in their particular cell lines. Norovirus, there has been a lot of work in the US to try and get a cell culture system up and running for that, but it's come across various issues and problems, and that's very, very expensive. So the whole cell culture side of things, if you were to go down that route, is more complicated, more time-consuming, and more expensive. But in terms of the detection of these, and because they don't grow on cheap agar plates, we have to do molecular detection. So molecular detection is always going to be a bit more expensive than your normal culturing. You need specific kits to be able to do that. You have to have very specific kits that can look for norovirus genogroup 1 and 2, and for hepatitis A virus, and for a sample process control virus, and you have to have various controls in your molecular detection as well. Plus, of course, you need the expertise to do that. You know, not everyone working in microbiological labs will know how to deal with viruses. So there's various extraction methods that you need to apply to be able to get the viruses off the surfaces of the foods. It's not a case of just putting a food in, incubating it, you know, mashing it up and getting the viruses to grow. It doesn't work the same way. So that's why it's a lot bit more expensive.
Thank you for that. We've all been thinking about viruses more perhaps than we've ever done in our whole lives. And as I say, everyone seems to be an expert. It's a really interesting question with all this extra knowledge that we have about COVID. With the large environmental pool of viruses that there are, I mean, they are everywhere, are we seeing the development of variants of concern? So with COVID, we've seen the Delta variant that's come around, or are these foodborne viruses, are they less susceptible to mutations? So if you looked at a coronavirus and a norovirus, is there a difference in the way that they mutate?
That's a good question. I'm not a molecular geneticist or you know, the biologist in that term, but so I probably Public Health England would have a better idea to answer that question. But in general, the genogroups 1 and 2 of norovirus and the groups of the hepatitis A virus have remained fairly stable. Obviously you get subgroups within those genogroups. So you will get various areas where they have outbreaks, like in Australia, in Sydney, for example, G217, I think that one is. So you will get various levels within those genogroups, which come up every now and again, which are variants, but they will still be caught within the general genogroups 1 and 2 of that human norovirus. Hope that makes sense.
Yeah, it does. And then one of the things I was thinking that COVID is an encapsulated virus and norovirus is unencapsulated. But if you look at how easier they are to clean, it seems counterintuitive to me that the encapsulated virus, like the coronavirus, is easier to clean than
Yes.
norovirus, which is unencapsulated.
Yeah, and that was, of course, there was a lot of knowledge going about, wasn't there, at the beginning of the COVID pandemic, with regards to the importance of washing hands with soap and water, because obviously the soap degrades the lipid layer of the SARS-CoV-2 virus fairly easily compared to norovirus. So yeah, it's all to do with that structure, as you say. It's because that has a lipid layer that protects the inner workings of the virus, if you like, that is a lot more susceptible to detergents and to alcohol than is norovirus. And yeah, we've seen from a couple of those outbreaks that we've mentioned there that the freezing of the virus can mean that it can travel around the world fairly happily and remain infectious for long periods of time. I mean, the really only effective way of disinfecting surfaces or to get rid of norovirus is a bleach-based disinfectant. And that's why, you know, obviously you don't want to be bleaching your vegetables or, you know, because there was that concern at the beginning of the SARS-CoV-2 pandemic as well, wasn't there? There was actually people bleaching their vegetables and fruits before eating it, which is obviously a big no-no. But you know, the control of norovirus is more difficult because of the fact that it's a lot more sturdy in the environment than SARS-CoV-2 is.
I've got some terrific questions. Here's another one. Thinking about what should you be testing? Do you think norovirus and hepatitis testing should be included in, and give some great examples? Fruit testing, hand swabs, food contact surface swabs, water testing. So what do you think? Are those the kind of things that you would be directing people if they're doing a sampling exercise and testing?
Yeah, I mean, you've got to think of where these viruses are coming from. They're coming from humans. Where's the primary contact between the humans and the food that is being consumed, which may be contaminated? Some of it's handlers' hands. We know that for sure. You know, the springtime, summertime, you know, the fields are full of people picking strawberries, raspberries in fields. You know, and you know, we tend to think of the UK as having very high standards of hygiene, which of course we do in the retail sector and all the rest of it. But, you know, in these fields, you might have one portaloo in the corner of a field, not great hand washing facilities. You know, you might have some hand sanitizer, but that won't kill off norovirus or hepatitis A. So yes, certainly in primary production, I think that is where there possibly should be more monitoring, because then you will have an idea of if the practices that are being performed in these areas where, you know, as we know, you know, basically we've got these workers who will put the fruit right into a punnet. It gets sold directly to the customer, and you're eating it after that person has picked the fruit. You know, obviously you're advised to wash it, but as we know, viruses stick very well to the surface of fruits and vegetables, so washing may not be completely effective. So, yep, irrigation water. There isn't any procedure for looking for viruses in there. I mean, sometimes they are checked for bacterial indicators, but bacterial indicators do not correlate with the presence of viruses and vice versa. So I think there could be more monitoring in place, and that in turn would give you a better idea of how you could potentially introduce control measures into the primary production and handling of these products.
It is amazing how we come into contact with these viruses, isn't it? I was going down to Whitstable next week, and what I love about Whitstable is the oysters there. So it looks like I just missed that.
Yeah.
One of my favourite products is from one of my most popular retailers. I love their Majola finest dates, and
Yeah.
just as I was going to buy some, I buy them every fortnight. We had a problem with that as well with hepatitis A. So these
Yes.
are real cases that seem to be
Yeah.
not some theoretical risks. These are
That's
real
it.
risks affecting the public.
Yes.
Well, Martin, look, we've come to the end of what was a really fascinating presentation for me. Great participation and great questions are coming in, and thank you for answering them. Look, it's just time for me just to thank you on everybody's behalf. It was a really great presentation, and I'm sure it will go viral, Martin. Is
Thank you
that
very much, Sterling.
joke you probably heard a
Yes,
thousand?
I've heard that one a good few times.
Viral. Look, if we didn't manage to answer your questions and people have asked that, you can contact Martin directly, and his details are on the screen now. So please, if we didn't get to your questions, there were so many of them, which is always a good sign, you can actually contact Martin. And another question that was asked, we'll be recording the webinar. So this is all recorded, including the Q&As as well. And this session will be available for everyone who's attended today via email and on our IFST webinar hub. So if there's some bits of gold in there that you might have missed, certainly all that data about the level of virus that will cause a reaction, I'm going to go back and look at that again. That seemed really, really interesting. Some reminders from the IFST. Look, if you have an interest in food safety, you can join the food safety section of the new IFST's community page on its website. So go to the community page, get into food safety, and you can engage in these type of conversations. Just another thing, perhaps look out for the recently published horizon scanning document. If you haven't seen that, that's really interesting from the IFST. And they've created their vision for the National Food Strategy. Both are really interesting reads. I'd recommend them. Also, if you've still got a need to know more about viruses, there's a foodborne virus information statement on the IFST website as well. Well worth a look at. And you can find links to these at the bottom of the survey, which is going to be launched after the webinar. So if you want this extra information, the links are on there. Hey, on behalf of the IFST, and in particular its food safety group, thank you all for attending. So it's great that you've joined us. Certainly the questions have made a major contribution to getting a better knowledge about foodborne viruses. And one last thank you. Martin, what a terrific job. Thank you so much. I've learned a lot. And goodbye, everybody, and stay safe. Goodbye.